one of our first speaker on the Flash talk, and I think it's Zoltan Fussy.
Yeah, I think so. I'm not sure how to share my share screen. Okay. Yeah. So hello everyone. I'm here to talk about the cryptic plastid of Euglena longa that was found to define a new type of non-photosynthetic plastids. As you probably know, then there are many lineages that acquired photosynthesis among eukaryotes, and there are at least as many eukaryotes that lost photosynthesis among in these lineages. So it seems to be a very frequent phenomenon. One of the the best known non-photosynthetic plastid is the Apicoplast. It's essential because it provides compounds to the host cell, and namely these are isoprenoids, tetrapyrols, and fatty acids. In striking contrast, there was almost nothing known about the cryptic plastid of Euglena longa, and previously we've shown that we have we've produced sufficient data to create or to make an in-depth analysis of the function of this plastid. So to cut the long story short, obviously there is no photosynthesis in Euglena longa, but surprisingly, we neither found the typical plastid of the Apicoplast, which is synthesis, isoprenoids, and fatty acids, and all these pathways localize in the cytosol. So there's a parallel pathways. There are parallel pathways that took over the function after the loss of photosynthesis. In comparison, we found evidence for glycolipid biosynthesis in the plastid of Euglena longa that's essential for membrane homeostasis of the plastid. We found evidence for the synthesis of two phytol derivatives, possibly involved in antioxidant activity, and the other one is not quite known. You can see further details about these in our paper that just came out, but I would like to concentrate on the last two, and this is the Redox relay system and the Kelvin cycle that are resident to this plastid. So the ferredoxin and thyrodoxin redox relay are functioning in line with the photosynthetic apparatus. They sense the level of NADPH in the cell, and depending on it, when the level is high, they activate downstream enzymes. So the ferredoxin thyrodoxin relay system in Euglena longa apparently derives from the system and is supported by the activity of the SOF assembly, which is an iron assembly system that assembles and delivers iron sulfur clusters to ferredoxin and ferredoxin thyrodoxin reductase. So indeed, phylogenetic analysis is consistent with the origin of the photosynthesis from green algae, and you can see that Euglena longa only has one of these enzymes. So what could be the function of the ferredoxin relay system? As I mentioned, there's an almost complete Kelvin-Benson cycle resident in the plastid of Euglena longa, and it seems that at least 5 enzymatic steps require an ferredoxin-dependent activation. And so this is readily visible also from the alignment, where you can see that Euglena longa always conserves the required residues for the activation. So we were thinking what could be the function of this whole thing, and one thing that really comes to mind is that this is not an isolated thing. So the pathway somehow cooperates with the cytosol, and it probably exchanges triose-phosphates with the cytosol through the triose-phosphate transporters. And when there is an accumulation of glyceraldehyde triphosphate, this has the effect that also NADPH is accumulated and activates the rest of the pathway. And why we think this happens is because of the stoichiometry of conversion of glyceraldehyde to 3-phosphoglycerate. And when you look at the equations, you can see that although the Calvin pathway consumes ATP in these reactions, it doesn't lead to accumulation of NADH. So we think that the reason why this mechanism has been preserved is that longa, since it survives on reduced carbon sources such as ethanol and lactate, it has to prevent the accumulation of the NADH or the reductive agents compounds in the cytosol. And this way, the Calvin cycle could function as a redox valve that would mitigate the effect of this accumulation through carbon fixation. So thank you for your attention, and of course my co-authors for their patience and grant money.